The broadcast is now starting. All attendees are in listen-only mode.
Hi everybody, my name is Ed Davis. I'm Senior Application Scientist at GeneCopoeia Incorporated in Rockville, Maryland. I want to thank you all for attending our webinar today on GeneCopoeia CRISPR sgRNA libraries for functional genomics. We really appreciate it. Before I get started, I want to just go over a few practical matters. First, you may ask me questions at any time, but to do so, you will need to type your questions in your chat box, which is located in your control panel on one side of the screen. You will not be able to speak vocally to me. If you cannot see your control panel, it might be hidden. In that case, look for a small strip of buttons on one side of the screen. One of the buttons will be an orange button with a white arrow pointing in one direction. If you click on that button, you'll then be able to see your control panel. Secondly, even though you can ask me questions at any time, for the purposes of keeping the talk flowing smoothly, I will wait until the end of the talk to answer your questions in the order in which they appear. And finally, please keep your chat box enabled because I will paste a couple of links for downloading a tech note or for attending other webinars into the chat box, and that way you'll be able to click on those links for either download or registration. So without any more to say, I'm going to go ahead and launch into our talk. All right, so I just first want to tell you a little bit about who we are at GeneCopoeia and what we're about. As you can see in the middle of this figure, GeneCopoeia was founded, and our primary product line is a large genome-wide collection of mammalian, mostly human and mouse ORF clones, open reading frame clones. And these are ready-to-express clones, primarily for mammalian cell culture that you can drop. They come ready to express, you can drop into your cell lines right away. But since then, we have branched out into various other product lines such as promoter clones, shRNA, proteins, and of course the product line related to what you're about to hear today, genome editing by CRISPR mostly, and some tailing. But the whole point of showing this slide is to tell you that in one way or another, most of these other product lines that we have are related in some way to our ORF clone product line. All right, but of course today's talk is about genome editing, so this slide shows a general overview of our genome editing products and services. At the most basic level, we do construct and clone construction and design, mainly of TALENs and CRISPRs for genome editing. However, we also have a number of services such as functional validation of the CRISPR clones, and also some more premium services such as the generation of stable cell lines or transgenic mice using TALEN and CRISPR technology. So here's an outline of today's seminar. First, I'm going to give you a brief introduction to functional genomics and CRISPR, and then I'm going to tell you about what CRISPR RNA libraries are. Then I'm going to go into a little bit of the development of previous sgRNA libraries and what our pathway and gene group specific libraries are, and and what the advantages are of our libraries over the previously developed libraries. All right, so let's start off with a discussion on functional genomics and CRISPR. So in the wake of the human genome sequencing projects, the biggest challenge stemming from that was to assign function to the sequences of the genome, especially all the genes and other functional units that have been identified. So getting the sequence information, as hard as it was one thing, but assigning function to these different elements is a completely different challenge. And so, the main questions people want to know is what are, promoter elements or transacting transcriptional factors that are involved in the region, and also what happens when you knock out a gene. Usually it's done in exon 1 or something as close as possible to an initiator ATG. People also want to know what you can do to other exons in the gene. This might involve exon modification for things like point mutation or correcting disease phenotypes. Or some people like to put functional tags on proteins so they can follow subcellular localization of a protein, for example. And it turns out that genome editing tools in general and CRISPR genome editing tools in particular are very useful for answering many of these questions and for accomplishing many of these goals. So the central focus of today's talk is related to CRISPR. So if you haven't heard about CRISPR, then what I can tell you is that it's an RNA-guided endonuclease that's used for widely used for genome editing technology. As shown in this figure, the yellow blob here represents Cas9 nuclease from Streptococcus pyogenes. It's been mammalian codon optimized for many applications, although it is used in other organisms. But this cannot function without the presence of what's known as a single guide RNA, which is a chimeric RNA that gives the nuclease target site specificity mediated through a 20 nucleotide target site. When these two come together, they find their target sites in the genome, and in the wild-type version of Cas9, or the standard version of Cas9, creates a double-strand break. There are some other mutants that create single-strand nicks, for example, but today we're just going to focus on the standard Cas9 that creates a site-specific double-strand break. When these double-strand breaks are created, cells have two main options for repairing these breaks. One is through homologous recombination, where you can knock in virtually any exogenous DNA sequence you want as long as there's homology surrounding the break. Or, and this is more closely related to what we're talking about today, there's a pathway known as non-homologous end-joining, or NHEJ, where the break is repaired by simply sticking the two free ends of the chromosome back together. This is an error-prone process which often leads to knockout mutations by frame shifting. And so it's this property that researchers have exploited in order to do readily perform gene knockouts. Here's essentially what happens with a knockout. If you look at, in this case from a published paper, there's a wild-type sequence in the EMX1 gene, and then you get, after CRISPR mutagenesis, you can isolate single clones in a cell line transfection experiment, and you'll see mostly small deletions. Occasionally, much less frequently, you do get small insertions. But again, if these are out-of-frame deletions, then these will almost always knock a gene out by frame shifting. So that is the type of technology that's being used for the CRISPR sgRNA libraries. So I mentioned that we have pathway and gene group specific sgRNA libraries. This slide is a general overview of that, and of course I will go into more detail later, but I just want to tell you up front what we have. We have 8 different libraries that are intended to knock genes out in various gene groups and pathways. Sorry, seven different groups. This is a list of the pre-made group here, okay, and we can also generate custom single-guide RNA libraries. They're available in a number of different formats. So you can receive these libraries either as bacterial stock, transfection-ready DNA, or lentiviral particles, because they're in lentiviral vectors. And they can be ordered in any of these formats either as pooled libraries or as individually arrayed libraries. Okay, so now that I've told you that, big question on this slide is, what are CRISPR sgRNA libraries? Well, first, there are collections of hundreds to thousands of plasmids that encode these target site-specific single-guide RNAs, also known as sgRNAs. So in the presence of Cas9, as I showed you earlier, they create double-strand breaks, or DSBs, that are intended to knock genes out. Naturally, since they encode many, since they target many genes, they're intended to knock out many genes simultaneously, and they can be designed to either target the entire genome or smaller groups of genes. So now I want to talk about briefly the applications for CRISPR sgRNA libraries. So there are a few main categories of applications that have previously been carried out by shRNA libraries, or short hairpin RNA libraries for RNA interference. The first application we'd like to talk about is drug target discovery, and all these references now are for shRNA libraries. Here's an example of one such library. You can also do reporter assays using a fluorescence marker, for example, on the cell surface and seeing if knockout causes a loss of that marker. There's also loss of function phenotypic screening. For example, you might be doing a soft agar assay to look at cancer cell invasiveness. So here's an example from this paper. And then there are others like drug target validation, etc., that I'll go over a little bit more later. So now again, pretty much all of these applications for sgRNA libraries can and have been carried out by shRNA libraries previously. So it might be natural to ask, well then why not just use shRNA libraries instead of CRISPR? Well, that's because CRISPR for these types of applications presents a number of advantages over using RNAi technology. For one thing, CRISPR makes a permanent change in the genetic code, so it's extremely stable. It's heritable, passed on down through through cell divisions. Both technologies do, which is not the case for RNAi, of course, does not change the genetic code. It acts on the RNA, not the chromosome. Both cause a change in expression level, but RNAi most notably causes what's known as a knockdown, or an incomplete elimination of gene expression, rather than a knockout, which is what CRISPR is intended to do. And in many cases, RNAi is sufficient to give you a phenotype or change your assay, cause you a change in your assay, but the fact that there is some expression left over can often confound your experiments. So it's a notable deficiency of RNAi, which CRISPR does not suffer from. In many cases, you can achieve a complete knockout of your gene. So now I want to talk about how CRISPR sgRNA libraries are used. Well, one of the important considerations, and this is also true of shRNA libraries, is that you need a good readout or phenotype or assay in order to best use these libraries. So in this slide, I'm going over two potential strategies for the use of single-guide RNA libraries. In part A, shows a pooled strategy where you use lentiviral transduction to infect a cell line with pooled sgRNA library lentiviral particles. So these pools will contain often hundreds to thousands of genes, like sgRNAs targeting genes, as I mentioned. And then once that is done, you can screen plates for a response to your phenotype of interest. And then in the pooled strategy, after a number of cell cycles, or applying, say, a drug of interest, or some other challenge, you then take the pooled cells, isolate DNA, and you can do PCR and sequencing to see which sgRNAs are represented or underrepresented that have caused your phenotype. And this is most often done on very large libraries using next-generation sequencing. In strategy B, we see the application of a single arrayed approach where each sgRNA is applied individually to cell line of interest. So then the cells are infected individually as individual sgRNAs to the cells, and then again you screen your plates for a response to a phenotype of interest. But then at the end, you can simply identify which sgRNAs are causing your effect in each well, because the sgRNA in each well is already known. So that's an advantage of the individually arrayed sgRNAs over the pooled strategy. All right, so what are the possible readouts? Well, I'm going to show some examples. Example number one is cell death. So let's say you want to identify novel drug targets, potential drug targets in a cell line of interest. Well, then you would expect that knocking out certain genes using CRISPR would actually kill the cells. So in this step, the first step is to infect your cell line with pooled sgRNA lentiviral particles. Alternatively, you can do a transfection with DNA. In step 2, after a few days of cell division, you prepare DNA from the mixed populations of living cells or surviving cells, and you can either perform Sanger sequencing on selected sgRNAs, like in a candidate approach, or you can perform deep sequencing, like next-generation sequencing, and analyze the representation of all the sgRNAs before and after infection. So in this scenario, sgRNAs that are underrepresented in the surviving cells represent candidates for genes necessary for a biological process of interest. And then, as in any kind of screen like this, you then test individual candidate sgRNAs in the same assay in order to validate your hits. In example readout number 2, we drug resistance, we actually have the opposite scenario. So again, you start off in step 1 by infecting your cell line with pooled sgRNAs. And then step 2, after cell division, you apply your drug of interest to the cells. And then in this case, you perform sequencing again, either by Sanger or next-generation sequencing. However, in this case, sgRNAs that are overrepresented in the pool represent candidates for genes necessary for the drug mechanism of action. And then again, once you've identified those initial candidates, you test individual candidates for validation. And finally, another readout you can do is a reporter assay. So let's say you have a GFP expression, a protein expressing with GFP in a fusion. And you're looking for a loss of that GFP, or vice versa, you can look for activation of the GFP signal. So again, you start off with infecting with the pool, and then in this case you might do FACS sorting to enrich for cells expressing the fluorescent reporter. And then after that you take the pool and do sequencing, as I mentioned before. But in this case, sgRNAs that are either under or overrepresented in the pool can represent candidates for genes necessary for your biological process that you're studying. And then of course at the end, once again, you test individual candidates for validation. All right, so now that I've given you that primer, let me tell you a little bit about the development of previous CRISPR libraries. There have actually been a few papers that have come out, but these are the two earliest examples from early last year. These are co-published in Science. One was from Eric Lander's lab, and the other is from Feng Zhang's lab. And in these examples, they use pools of lentiviral delivered sgRNAs for large-scale forward mutagenesis screens in human cells. In the Wang et al. Paper, they had a pool of sgRNAs targeting 7,300 genes, and they were looking for drug resistance using 6-glyothonosine drug selection. And after drug selection and next-gen sequencing, they found that there was a vast enrichment for sgRNAs that are involved in the mismatch repair pathway, but not other DNA repair pathway genes. And this was a hypothesis test, and these results validated this hypothesis. In an even larger screen, it's not shown here, but in the Feng Zhang's library screen, they targeted 18,000 genes with a pool of sgRNAs, and they were looking for PLX drug resistance in a melanoma cell line. And in this case, they compared the PLX-treated cells with vehicle-treated cells, and they found a change in the representation of the sgRNAs in these pooled cells. When they compared the pooled and the vehicle with the drug-treated cell lines, they saw a number of candidates that were overrepresented in the pool. They tested the 6 best candidates in this case in validation assays, and they found that all 6 of those were validated in this assay for PLX resistance. So now these were great results. They showed that the technology is very useful for large-scale screening. However, now I'm going to tell you about our recently developed pathogen gene group-specific sgRNA libraries and what the specific advantages are over these previous libraries. So this is a slide I showed before showing our seven different pre-made, pre-designed sgRNA libraries. These are much smaller than genome-wide. They're pathway and gene group specific. I also mentioned that you can custom order virtually any library you want, and we can make it for you, and they're available in a number of array formats. So here's a list of the advantages of the GeneCopoeia sgRNA libraries over the previously developed libraries. First, these sgRNA plasmids are individually constructed and cultured in E. coli before pooling. And the main benefit of this strategy is that it makes possible for using these sgRNAs either in pools or as individual sgRNAs. The previous libraries that I discussed were actually constructed right from the beginning as pools, so it's impossible to separate them out as individuals. Secondly, the pools are limited to 150 sgRNAs each, and the main advantage of this is that it ensures excellent representation of each sgRNA, which is a challenge in much larger scale libraries. Also, each sgRNA is sequence verified, which ensures the high quality of each of these clones. And finally, the small library sizes vastly reduce the time and cost of screening. So, in other words, when you screen, there's no need to use next-generation sequencing in order to look for over or under representation of sgRNAs, because you can simply test them by Sanger sequencing, and you can get the results earlier and at a lower cost. So here's a schematic of the library pooling strategy. First, each plasmid expressing the individual sgRNAs are constructed separately and sequenced. The plasmids are then individually used to transform E. coli, as shown here. And then it is only at this point that the E. coli cultures are pooled. And then from these pools, we prepare plasmid DNA, which we can ship out directly, or we take this pooled plasmid DNA to produce pooled lentiviral particles. The libraries are also available as two pools, pools A and pool B, and these are redundant pools that target the same genes but with different sgRNAs. And as I mentioned, the number of sgRNAs in each pool is limited to 150. This is the vector backbone that we used. Again, I mentioned that it's in a lentiviral vector backbone. We also refer to this as a dual-use vector backbone because it can be used either for transfection or transduction. The backbone contains all the necessary elements for third-generation lentiviral packaging systems, and it contains a puro-selectable marker for stable integration in the genome, selection of stable integration in the genome, and M-cherry fluorescent reporter. So each sgRNA is cloned into this lentiviral vector backbone. Now, there are a number of considerations that we recommend for using these sgRNA libraries, and one of them is that it is best to start with a Cas9 expressing stable cell line that you would then use to transduce with your sgRNA libraries. In one particular option, we have lentiviral clones, lentiviral-based vector clones that carry the Cas9 nuclease, and you can use these to stably integrate the Cas9 nuclease. This is a random integration, though, and so if you don't like random integration, we have another option in which we can integrate the Cas9 nuclease at the AAVS1 site on human chromosome 19. AAVS1 is also known as the safe harbor site in humans, and that's because insertion at that locus has no known adverse effect or phenotype on cells. And so if you use this and we have some kits for knocking these constructs into the safe harbor site, if you use these markers, these plasmids come with either hygro or neomycin resistance, so they're fully compatible with the library plasmids. And so this gives you the option to either purchase a pre-made Cas9 expressing stable cell line from us. We have a few of those. You can also order a custom Cas9 expressing cell line of your choice, or you can purchase these donor plasmids for do-it-yourself transfection into your cell line of choice. All right, so now I'm going to break out from the PowerPoint, and I'm going to go to my web browser to show you how you can easily order these sgRNA libraries from us. Okay, so I will exit that temporarily and then open up our web browser. This is the GeneCopoeia Human sgRNA Library web page, and as you can see, there's a table listing each of the sgRNA libraries. So first you need to start by choosing your delivery format. So let's say I want to choose lentiviral particles. And so you get a message saying that each tube contains 2.5 times 10 to the 6 transduction units in 25 microliters. And this is usually enough for a few, maybe 3 to 4 transductions, depending on the MOI you need to use for your cell line. And of course I mentioned that you can order either bacterial stocks or DNA. Okay, and so then it's very simple. Once you've picked your format, you then select the library that you want, and then you just add to the shopping cart and then check out. So it's very easy to order right on our website. If you want a custom library, though, you need to contact us for a custom quote. And also, I meant to mention that the website ordering is for the pooled sgRNA libraries. So if you want the individual RAID format, again, you need to request a custom quote. All right, so let's go back to the PowerPoint. Okay. So now let me just tell you that we have a number of related products and services that are meant to accompany the sgRNA library products. Again, I mentioned before pre-made Cas9 expressing stable cell lines. So we have a few of these on our website, and we are making more of them. But essentially, these are stable cell lines with constitutively or inducibly expressing Cas9. I mentioned we have pre-made lines, or you can have us integrate this sgRNA or Cas9 donor construct in your cell line of choice. The clones are available for do-it-yourself stable cell line creation. Cas9 is integrated safe harbor locus for good consistent expression and insertion without consequences. And these stable cell lines or the vector are ideal for sgRNA library screening or validation. In addition, I mentioned that the donor clone that expresses Cas9 is expressed from the human AAVS1 safe harbor site. And so we have a number of kits that are that you can use to do that knock-in at the safe harbor site. I already mentioned the benefits of using a safe harbor. So the kits come with the CRISPR tailent reagents you need to do the knock-in, and with a controlled donor clone, and then you would just order the version of the kit that has no donor along with the Cas9 expressing donor clone. We also have a number of safe harbor kits with knock-in ORF clones, where you can order any human or mouse ORF of your liking, and we can clone it into the safe harbor locus for you, and then you can use that to knock in an ORF in your cell line. Okay, and this is just another example of the safe harbor knock-in clones. In addition, we have lentiviral production service and packaging kits. So if you order the plasmids from us and want to do your own packaging, you can order the packaging kits from us. You can also have us do the lentiviral production, especially for custom libraries. So we package the lentiviral-based clones into ready-to-use particles. These are ideal for the CRISPR lentiviral clones, and we can either package individual CRISPR clones or our own libraries that I mentioned before, or we can even package DNA into particles from outside libraries as well. All right, so now I'm nearing the end of the talk, and I just wanted to tell you that if you want to know a little bit more or want some reading material, we recently released a technical note on applications for CRISPR single guide RNA libraries. You can download this straight from our website. This is the download link, but I'm not going to make you, I expect for you to copy this link yourself. Instead, I'm going to copy it and paste it into the chat box as I mentioned before. Okay, so I just sent it on, and so all you need to do to download this technical note is to click on the link in the chat box. All right, so now I'm at the end, and I just want to briefly summarize. First, I told you about CRISPR sgRNA libraries, which have tremendous potential for functional genomic screening. I discussed some applications for CRISPR sgRNA libraries, which include novel drug target discovery, drug target validation, phenotypic screening, reporter assays, and more. I told you about the advantages of the GeneCopoeia sgRNA libraries over the previous developed libraries, which include sequence validation, individual clone construction, and individual culturing in E. coli. And finally, I told you about how GeneCopoeia CRISPR sgRNA libraries are available in a range of formats and have many powerful companion products and services. All right, so before I take questions, I also want to tell you about some upcoming webinars we have. We give webinars on CRISPR and genome editing fairly often. So the next GeneCopoeia genome editing webinar is a general discussion of genome editing, and this will take place on Wednesday, May 13th at 9 o'clock in the morning. That's Eastern Standard Time. And this is the registration link, but again, I'm going to copy and paste this registration link into the chat box. Okay, I just sent that one along. And then there's another upcoming webinar, which will be on our GeneCopoeia genome editing stable cell line services. We at GeneCopoeia can construct, as I mentioned before, genome-edited stable cell lines using virtually any cell line of choice and any genome editing application of choice. This will occur on Wednesday, May 27th at noon Eastern Standard Time. And so I will also copy and paste that registration link into the chat box. And if you're not seeing these additional links, it probably means you need to scroll down. Hang on a second, I lost my control panel. Okay, and I just sent that link along. Okay, so with that, I'm going to stop. And again, I want to thank you all for attending the webinar. I hope it was informative. So now I'm going to take questions. So the first question is, if 2.5 million lentiviral particles or transduction units is not enough, can I get more? Well, yes, of course you can get more. We do lots of lentiviral packaging reactions for people, so that is no problem. You would just need a custom quote for that. Okay, next question is, can I just order individual sgRNAs from your libraries instead of the whole library? The answer is yes. Again, you would just need to call us for a quote. And I see one more question here. Can we get the gene lists of that are in the libraries? Yes, you can. Those gene lists are available on our website. You just need to first create an account. All right, so are there any other questions? Anybody out there? Okay. All right, well, if there are no more questions, then again, I want to thank you for your attention and to have a great day. Bye.